So the second program is going to be more focused on transcriptomic data analysis, or RNA-seq. And we're going to talk about bulk RNA-seq, when you extract your RNA from the whole tissue and you kind of analyze levels of gene expression and compare them between different groups or creating different contrasts. But we'll also talk about single cell RNA-seq, when you extract RNA with a barcode that can make it specific to a cell. And so this is an exciting field with a lot of new methods that are becoming available. Not all of them are simple to use, and in many of them, you need to use them knowing how they work, so how the data is prepared, but also what are some of the standard procedures and considerations about the assumptions that we have and about the conclusions that we will have from our analysis. So we will talk about first of all, the biology of transcription as a proxy for translation. How can we actually use the abundance, the quantity of RNA of a specific type to infer how this will turn into a specific protein? So this is kind of the most common use case, right? We want to identify some functional state, and we say that the mRNA is the proxy that allows us to accurately identify which genes are actually, which protein coding genes are being expressed. And that is why we think this leads to some functional change. Now, there are several different methods to measure and quantify RNA, but we will exclusively focus on next generation sequencing. And for next generation sequencing, there are several levels that you can kind of use that data to analyze what's going on. So one of them is just to look at genes, and these are protein coding genes. Another one is to look at alternative splicing events, where you have multiple exons that connect together, and you would like to understand how that leads to different transcripts or different isoforms, so to say, of the same gene. And so that oftentimes is a very useful technique to be able to understand what is actually going on, because not in every case, loss of function is associated with a low level of expression for a particular gene. So we'll talk about the differences between gene and transcript level analysis, but we'll also talk about how to look at that detail by making sure that we are actually developing a transcriptome from our own data, and only then going through the process of annotation. Now, we will try to apply this in the context of a biomedical research project. And so we'll talk about how bioinformatics allows us to essentially connect clinical and omics data, how it allows us to address questions around diagnostics and application of therapeutics. So we will kind of extend our understanding of this expression of genes, isoforms, and proteins, translation into proteins, to then ask more biomedical type questions from the data that we will explore. The program will have several different sessions. We'll start from just the basic logic of what I explained. How is that processing done? And what are the important steps in that pipeline? And the important steps are going to be how the data is prepared with minimal artifacts, so that our conclusions about it at the very end are actually reliable, something that we can trust. And so we'll talk about different methods of data pre-processing or cleaning. We'll talk about mapping. How can we map on the transcriptome, on the genome? How do we actually identify those alternative splicing events, etc.? And then we'll talk about how can that data be quantified and used to represent a difference between conditions. So in the most simple experiment, you will have several replicates in one condition, several replicates in another condition, and because they have been kind of produced in a controlled way, you can make biological interpretation based on these differentially expressed genes. And we'll talk about plotting, volcano plots, and things like that, but also about downstream interpretation. How do we connect those differentially expressed genes with gene sets, like pathways, gene ontologies, etc.? And how do we make sense of what's actually going on? Now, to do that, we will have to cover all of the steps from the basic alignment, as you see here, with the reads aligned to the reference genome, to then quantification, analysis of variance, and understanding of whether it is the right approach to be able to compare multiple conditions, and how do we actually go about more complex data sets where instead of just two contrasts, you all of a sudden have many. How can you analyze that kind of data? And that is, again, bringing us back to that question of data mining and classification, really being able to work with larger and more complex data sets and extract meaningful insights by using nonlinear methods of machine learning and feature selection. So then we'll also talk about single-cell transcriptomics. So an exciting technique becoming very, very popular. Many cores are now adopting 10x genomics and other machines to be able to produce single-cell data. Question is, what can we do with it? And how do we do it so that we are in control of that analysis and we know how to ask biological questions and actually get answers to those? So to do that, we will talk about typical approaches. So the standard output being the UMAP or the t-SNE presents us with a clearer picture that unfortunately is not trivial to explain, because it really contains a lot of information that has been adapted to a particular hyperplane, and it's difficult to connect back to biology. So we will look at how we can use such methods of dimensionality reduction to start asking biological question, going back to the statistical comparisons that we've explored in the bulk RNA-seq course, and how do we then can also ask additional questions about, for example, pseudotime. How can we track trajectories where gene expression levels actually represent a continuous process? Then we will look at the full pipeline again, from understanding of what is in our data to then being able to ask specific questions, and then all the way to downstream interpretation. Now, all of these are going to be given in the context of several different project examples. And we'll talk about patient-derived xenograft models, where we will compare tumor and the microenvironment. We will talk about immune cells and ability to understand different subtypes of cancer. We'll talk about macrophages, how the complex immune responses could actually be both detrimental and supportive of tumor development. And we'll also talk about early stage cancer detection, like biomarkers in exosomes. So a number of different types of projects that we will look at. We'll offer also a collection of curated data sets that you can kind of try to apply some of the methods that we went over in the program to something that is more relevant to maybe your research interests. And we did call this transcriptomics for chronic diseases. So those are the types of examples that we're going to use. But if you would like to then use it to a data set of your own, that is also going to be possible. And we have several ways that you can do that, including talking to someone that can guide you through that process, discuss your project with you, and help you kind of arrive at those results.